This video is all about different kinds of media that we will be using in lab and a bit of history about how we got to this point in microbiology lab in terms of how we got to using certain types of media, which is what you grow different bacterial cultures on or in, okay? So Robert Koch, remember, he was in a competition with Pasteur to try and prove that certain microorganisms cause certain diseases. Okay, and he was looking to prove that Bacillus anthracis specifically causes anthrax. And it turns out he won this race and eventually led to the germ theory of disease, which again is that a specific microbe causes a specific disease, okay? And one reason he was able to win this race with Pasteur was because his people in his lab figured out how to make media that would allow organisms to grow in the lab. Because look at Koch's postulates. We did talk about this a little bit in the Intro to Micro series. So the first postulate is that the bacteria must be present in every case of the disease, right? If Bacillus anthracis causes anthrax, every person that has anthrax better be infected by Bacillus anthracis. Because if they're not, clearly that organism is not causing the disease. So the next step is that you must be able to isolate the bacteria from the infected organism, okay? And then once you isolate that organism from the infected entity, you can grow that organism, that microbe, in the lab. So that way we can grow lots of it, and we can then use that bacteria to go infect healthy hosts. And then those healthy hosts should become sick because we've infected them with the pathogen. So key to this process of associating a microbe with disease was being able to grow the microbe in the lab. And they were having one heck of a time doing that, okay? So originally bacteria were grown mainly using broth. But the problem with that is bacterial growth turns the liquid broth cloudy or turbid. But when you look at a test tube and you see that it's turbid, you can't say for sure that only E. coli is growing in there, or maybe only Bacillus anthracis is growing in there, because the two organisms are going to grow identically. Is that a word? They're going to grow the same in the broth media. They're going to turn it cloudy. So there's no way of knowing if your broth culture is actually contaminated and has multiple organisms growing in it, or if your culture is a pure culture, which means it only has one organism growing in it, the organism you're trying to study. So Koch needed to grow the organisms on a solid media because it turns out different species grow differently on solid media, okay? And you're gonna learn a lot more about that in upcoming videos. But on an agar plate, Staphylococcus aureus grows a goldish color. It forms little gold circular colonies. Colonies are a mass of cells that we can visibly see with the naked eye, whereas Micrococcus luteus grows bright yellow. E. coli grows beige. And so you don't need to memorize that information for now, but I'm just letting you know that by growing things on a solid media, you can look at how they grow to differentiate different organisms. Or say, look, all these colonies look exactly the same. Therefore, there's only one species growing on this plate. So there's no contamination, okay? So with this attempt to grow or to develop and use solid media, he tried things like potato slices and egg whites. But the problem is there are set nutrients in a potato slice, right? You can't really add additional nutrients to it. You can't subtract nutrients from a potato slice or egg, right? It is what it is. And so only some organisms would grow on potato slices or egg. But for those bacteria that had more picky nutrient requirements, they wouldn't be able to grow on these solid media, okay? So he needed to be able to change up the nutrients available in that solid media so that anything could grow, whatever it was he was studying. So Julius Petrie was working with Koch, and he actually gets credit for developing the glass Petri dish. And by having the lid on the Petri dish, it was preventing contaminants in the air from, contaminating the solid media. So this glass Petri dish was super helpful, step in the right direction, but now Koch needed the medium that could go into it, right? So one of the main ingredients that Koch was using was gelatin, right? Which is what we use in Jell-O. But there were a few big flaws with gelatin. Gelatin has a pretty low melting point. So when they were putting these gelatin plates in an incubator at 37 degrees Celsius, which is about our body temperature, they were starting to melt, right? Well, you can't grow bacteria on a melting surface, right? Counterproductive. Second, gelatin is a protein, and bacteria like protein, right? They need it. So they were actually metabolizing it. And then the media would become a liquid. So gelatin was not the solution that we needed. They needed. Well, turns out Walter Hesse worked in the microbiology lab with Koch. He really hired all the right people. And he was an illustrator, and he also did some science in the lab, but his wife worked in the kitchen, but she also helped make the media, because a lot of times they would use like a chicken broth or a beef broth to grow cultures in. So she helped make the media and do other basic like lab tech stuff like cleaning glassware. And she said, well, if gelatin isn't working, let's use agar. And that was the key to success, because agar has a much higher melting point and it has no nutritional value to the bacteria. Okay. And we still use agar to this day. But old Koch, did he give her credit? No. But we know the truth. But y'all, agar has another really, really helpful property, and that is that it is a solid at like room temperature, which is when we're working with it. But at really high temperatures, it's a liquid. And so this allows us to essentially create the different media that you see here, but well, specifically the solid media, the right three images, because agar is simply a solidifying agent. That is it. Again, it is not a source of nutrition. It is just a solidifying agent. But what happens is when we're making all of this media in the lab, we put all the different nutritional powders and things into a really large flask. We add water and we mix it all up. And then we add powder agar that is a powder. And again, we mix it all up and then we stick it in an autoclave. And let me go to that slide real quick. We'll come back to this one. We put it in an autoclave, which sterilizes the media, which means it kills any living thing, including endospores. And so the agar reaches 121 degrees Celsius at 15 PSI, which is a lot of pressure, for 20 minutes. So essentially it is creating superheated steam. And these conditions will kill, like I said, any living thing. It reaches sterilization, okay? So once the 20 minutes have passed, right, the pressure decreases and eventually we can open the autoclave carefully because a ton of really hot steam comes out. And the liquid in the flask is still bubbling. It's crazy hot. You have to use these big hand warmers to make sure you don't burn yourself. But you let the media cool for a little bit. But then because it's still in liquid form, you can pour it into these plastic Petri dishes, or you can pour it into glass test tubes. And then as it cools over time, takes anywhere from about 15 minutes with agar plates to maybe even a couple hours with some of the test tube media. But as it cools, it does solidify. Okay, so agar, being a liquid at really high temperatures, gives us the flexibility and the ease of pouring this nutritious medium into test tubes or agar plates. So it makes making media a lot easier. But then again, as it cools, it solidifies. So again, agar is a solidifying agent. So when we're looking at these 4 types of media, we have one that is a liquid, and that is a broth. So there is no agar in a broth, right? Because agar is a solidifying agent. Broth is liquid, not a solid. So there's no agar in a broth. An agar plate has agar, and so does the agar slant and the agar deep. So how is a slant different than a deep? Well, once we pour that boiling hot liquid that comes out of the autoclave, which contains the agar, right, liquid at high temperatures, we'll pour them into the test tubes. And then for the slant, you literally just take your test tube and you balance it at an angle so that as it solidifies, the media solidifies at an angle on a slant. So the slant is advantageous to a deep because it has more surface area and you can grow a lot of bacteria in a little space. So when I make stock cultures, so like I don't want to have to grow up E. coli every single week. So I can make stock cultures that I can use for several weeks and I just grow them on a slant. So I have a lot of E. coli on a slant that I can easily allocate to you all in broth cultures or whatever I need. So slants are nice because it's a small test tube. It's easy to store, but I can grow a lot of bacteria on it. The deep literally fills up normally 2/3 of the test tube, and it's just completely solid. And so we don't use an inoculating loop, which is the main tool that we use in the lab. You'll see a picture of it on the next video. We actually have to use what's called an inoculating needle, and you stab the agar deep. So we don't use the deeps very often, but it is nice because we use it for motility and for oxygen requirements because there's less oxygen at the bottom of this solid tube of media and there's more oxygen at the top. So when you stab the agar with an organism, if it tends to grow near the top of the tube, that means it requires a lot of oxygen. If it grows at the bottom of the tube, it does not require oxygen. And then there's options in between. Similarly, with motility, we can stab that agar and organisms that have flagella are able to swim away from that stab line where you introduced the bacteria. But organisms that aren't motile are stuck in that stab line where you put them because they can't swim away. So there's a couple of uses for the deeps, but we won't use that media all that often. So we already talked about autoclaves. I do want to point out, though, that a lot of times people think that boiling water or media reaches sterilization. And remember, sterilization means it kills every living thing, including endospores. Well, boiling temperatures are 100 degrees Celsius, and that is not sufficient to kill most things like thermophilic bacteria or endospores. And so boiling alone is not sufficient to kill everything. So we do use an autoclave in the lab to ensure our media is sterile. So what I really want you to take away from this slide is look on the 2 broth cultures on the left. Notice on the left test tube, you can clearly see the black color of the tabletop behind that tube. Sterile media, it has like a goldish color to it, but it's see-through. You can hold it up to the light and see straight through it, okay? So that's how you know media is sterile. There's nothing growing in it because it's not cloudy, okay? It's, again, you can just see right through it. On the right, notice how cloudy or turbid that culture is. That's how you know something is growing inside that culture, okay? So if I were to give you guys sterile broth culture and said, hey, I want you to put some E. coli in this for whatever purpose. If I handed you something saying it was sterile, but it actually was really turbid and cloudy, you should say, no, Chelsea, this one is contaminated, and we wouldn't use it, okay? On the right, you can see what contamination looks like. So this plate, sometimes when we're pouring media, if for whatever reason you leave the lid off too long or maybe you just accidentally sneeze while the lid of the Petri dish is off, you can contaminate it. Okay, it doesn't happen all that often, but it's possible. And so you always want to inspect your media before you use it. And so those three circles there, those are called colonies. Colonies grow on agar plates. And so essentially what happens here is an individual cell landed where each of those colonies are. So 3 single cells were originally gained access to this plate. And over time, about every 20 minutes, those cells would divide. So you go from 1 cell to 2 in about 20 minutes or so. And then you go from 2 to 4, 4 to 8, which we still can't see 8 cells with the naked eye, right? They're too tiny. But once 24 to 48 hours have passed and they've gone through many, many, many rounds of division, you'll have millions of cells deposited in that same area. And so that mass of cells is what we call a colony. Okay, so each colony arose from a single cell undergoing hundreds and hundreds and hundreds of rounds of cell division to make millions of cells in that one location that we can see. So that's a colony. So when E. coli grows on an agar plate, its colonies will look different than Staphylococcus aureus or Salmonella. Okay, and so we're going to talk later in the semester about how we describe colonies and how that helps us identify what's actually on the plate. All right, y'all, that is the end of this video. Let me know if you have any questions. Make sure that you do answer the questions at the end of these handouts. And if you have any questions, please let me know.